Hi, my name is Weixiang. I'll go straight into the talk. So I have presented this talk earlier at the Bodian Consortium meeting towards the end of last year. But so for those of you who have seen this already, hopefully I can add more information. Otherwise, the topic of today's talk is mapping complex cell-phase dynamics using what we call the cell-phase cell phylogeny. So I want to, let me see if my slides can advance. Okay. So I want to start this talk by putting out this question of how do we map the developmental history of complex differentiated cell types, which I think is a fundamental question that people ask in the field of developmental biology. So the first line of research that tries to address this question has to do and goes along the line of time course single cell omics, which I think most of the people probably are very familiar with. So as an example, this paper in Nature in 2019 did time course single cell RNA-seq from the embryonic day 6.5 to embryonic day 8.5, and targeting to study this duration of mouse gastrulation, right? So with this type of study, you can get an idea of which cell types are present at different stages of the development. And but I want to point out that with this kind of approach, you do have to sample very densely at these stages, and as the embryo do tend to change very rapidly during these times. And but these data provide you with a kind of a descriptive model of the process, right? And later in 2021, with some more careful modeling, staging the embryo and fitting what they call flow model, you can start to say something about dynamics, that is change between the cell types and how they occur over the same period of time. So we're a big fan of this work from the Masons-Wig et al. From 2021, right? But fundamentally though, this type of approach has one major shortcoming, which is that the embryo that you look at from the different time points are actually different embryos. The limitation of the current technology is such that one doesn't actually get to follow these embryos through time. So the last axis in the Bodian Consortium, where we try to look at the time axis, you can argue that cannot be completely addressed by this type of approach. Whatever you get is kind of an inference in between the time points, however densely you actually sample, right? But also, as this review paper was pointing out, even if you're modeling, even if you're doing a really good job with the modeling, there still are cases that one can get misleading conclusion by only looking at the state space itself. For example, there could be lineage restriction that doesn't really appear depending on which state space that you're looking at. And there are cases where the same cellular state can be reached following completely different developmental trajectory, such as being illustrated in these two examples here. So I would like to discuss an alternative line of thought for this problem, which brings us to the second approach that we will focus on today, that we call single cell lineage barcodes. So traditionally, developmental biologists have been using these lineage tracing techniques to understand the fate of the single cell of a progenitor cell. In this example, a random homologous recombination event labels all progenies of a single cell, right? And by histochemically labeling the progenies of these cells and observing the end fate that they end up becoming, one can really start to understand whether progenitor populations were present when the progenitor cells got labeled. This is one very nice example from 2009. So along this line of thought, taking this further in a sort of a more modern approach, fully taking advantage of the advancement in sequencing technologies, we reach to where we are now, which is what we call the lineage barcoding. You can think of it as a multiplex way of the traditional lineage tracing. So lineage barcodes are loci in the cell that gradually accumulate mutation during the development. At different stages of the development, each cell ends up with a set of mutations. And the mutations is heritable, so they get inherited to the other cells once the cell divides. When you collect the cells at a given time points and look at these barcodes, the more similar, more closely lineagely, cells that are more closely related in lineage share more similar barcodes. and vice versa, right? So at a given time point, by sequencing lineage barcodes in single cells, one can obtain an array of mutations in a number of cells in the form of a character matrix that is being shown here. In this matrix, the black represents a cell that is unmutated, and the color represents different mutations, right? So instead of in the traditional way where you get information about a single clone, by using these character matrix, modeling the mutagenesis process, and applying phylogenetic inference approaches, we can get to not just a single clone, but what we call timescale phylogeny. So here, by timescale, we mean that the branch length along these phylogenetic tree corresponds to actual time in between cell divisions. And you can do that because these mutations happen at relatively constant rates through time. And for each cell, I want to point out that we also know when we sequence them at one time point, we also know their corresponding cell types. For example, in this study from the last year, they also used a single cell somatic mutation to reconstruct the phylogeny for up to 10 cell divisions in human development. However, you can ask yourself here, by looking at this type of phylogeny, what information can we actually learn about the development? And it turns out it's not immediately obvious. As one simple example, you can see these blue cell types that are of the same terminal fate can appear very far away on the phylogenetic tree. But what we're actually really interested in getting is actually how individual cells go through these progenitor states during development to actually reach their terminal fate. So ideally, we want to get a map that looks somewhere like this instead of that is in this format, right? So this map will tell us what are the actual state transitions, state changes through time that have to happen for the development to occur. And turns out the reason for that is in that in higher organisms such as human and mouse, it's not an individual cell that governs the developmental process, like in single organisms such as C. elegans. So it is a number of progenitor states that governs this process. And in order to make complex organisms these progenitor states, there needs to be a large population of these progenitor states in higher organisms. So, which brings us to the main portion of this talk. So to address this problem, we have developed a method for reconstructing this type of map that we call progenitor state dynamics from the cell phylogeny. And we call this approach an I-space. So from what we think is a sparse sampling of the division history, given the number of cells that is sequenced, we hope to reach to a dynamics of the lineage progression during development, which is the goal here. And this was published late last year. So I will briefly give an overview of our algorithm to give you an intuition how it works. So our algorithm called I-space consists of two steps. And actually, the first step is called phase. And the second step we call the I step. So in the phase step, we reconstruct what we call the phase map topology, which is a hierarchy of progenitor states with different potency levels that is defined relative to the terminal cell types that were collected. And in the second step, that is called ICE, we reconstruct the time dynamics for each of these individual states that were identified in the previous step. In particular, we estimate the timing, size, and bias of these progenitor states. The phase algorithm tries to identify the latest time along this timescale phylogeny before a pair of terminal fate has separated. So in this hypothetical example, the highlighted node here in red denotes the places along these phylogenies where the T4 and T5 fate has separated with one another. In total, there are 5 total terminal cell types that are being shown here. So we first annotate each node with observed fate. We identify the fate separation, and then we look at where they occur along time axis. So across the entire phylogeny, many such phases for all pairs of terminal fates are computed. Here, 2 pairs of separation is being shown. And when you organize these distances in a matrix, you get a matrix that looks somewhat like this. And the value on the matrix is what we call the phase distance, that is phase separation distance. And the longer the phase separation distance, the earlier in development two fate has separated with one another, right? And by applying hierarchical clustering, we get to this reconstructed topology. So in our paper, one of our argument is that the key point in successfully using these phylogeny data is using what we call phylogenetic time of the progenitor cells to reconstruct the order of lineage segregation. And the figure here shows that compared to the previous method, which does not carefully model the mutagenesis process and use the phylogenetic time, we get to a much more accurate reconstruction in terms of the order of these progenitor states, or in terms of the order of these phase separation. So this figure here is showing the reconstruction error compared to our methods in some previous methods. And on the x-axis, we're showing the imbalance of the topology. And our conclusion was that when the imbalanceness of the topology is high, there can be a bias that occurs with a traditional method. So to recap the last part, the result of topology inference is a series of bifurcation along the tree, where along each bifurcation, the progenitor cell differentiate and lose certain potencies. Within this type topology, we call each of these bifurcation an inferred progenitor state, which is defined by its potency, that is the terminal cell type it can produce. So in the next step, which is the I step, we move on to estimate the dynamics parameter for each of these progenitor states. So the three dynamics parameter will be summarizing into the commitment time, which we know from the phylogenetic time. That is when the cell starts to lose potency and commit to downstream phase. The population sizes, which is how many cells were there right before the cell has committed. And the commitment bias, which is the fraction of progenitor cell that commits to one phase versus the other. So the ICE algorithm starts by assigning progenitor states to internal nodes of the cell phylogeny. And this is done by taking into account the overall topology that has been reconstructed. So instead of assigning the set of terminal cells that can produce, we assign one of the progenitor states that were inferred to each node in the phylogeny. And this provides, after this annotation, and when we look at the phylogeny again, what we see is a series of lineage progression along the phylogeny. And this figure shows the assignment over the entire phylogeny. And the assigned state then defined, and with this assigned state, we can see that the phase commitment happens when the progenitor cells change state. These events are what we call inferred commitment events, and hence the name of this algorithm. So in order to estimate the commitment time, all we need to do is to look at the phylogenetic time of these ICEs that were identified. So which brings us to one of the other contributions in this work that were done, which is that we pointed out the importance of sampling in designing these lineage barcoding experiments. Unlike traditional lineage tracing experiments, which I showed earlier, where the final fate of a progenitor cells is almost always observed due to the limitation in the number of cells that can be sequenced, whether depending on the technological reason or financial reason. For some progenitor cells, one can easily miss their progenies of certain terminal fate and hence misassign their potency, which really creates a bias in terms of all the estimates that were discussed previously, right? So in this example, the cell is actually capable of T1 through T4 fate, but because of the limited amount of sampling that one can do, depending on which stage was sampled and how many cells were sampled, you can misassign it to a less potent state in this way. And to show this, we used a simulation study to show that the estimates are only accurate when the states are being well sampled. So here, the well sampled means that the large majority of the progenitor state, the progenies of a progenitor cell are being sampled. Otherwise, the progenitor state is being undersampled. And this figure is showing that bias can occur in terms for the estimation of progenitor time when this is the case. So in this figure, x-axis is the true commitment time we know in simulation, y-axis is the inferred commitment time that we got from the algorithm. And the red color are the ones that are being undersampled. So next, after inferring the progenitor after inferring the commitment time for the progenitor state, we go on to infer the progenitor population size and commitment bias. And it's really a very similar idea, which is just to look at these ice distributions, right? So in here, we're showing a zoom in the view along along the commitment events of two different progenitor states. And by counting the nodes that are upstream and downstream of these commitment events, we can get an estimate of the population size and commitment bias. And our results show essentially similarly for the well-sampled progenitor state, we got very good results, but the undersampling state really struggled, whether you're looking at the population size or commitment bias. So in summary, what we discussed today is how to use cell phylogeny inferred from lineage barcodes to learn about both the shape of the fate map topology and also to reconstruct the dynamics of the progenitor state. Before I go to the acknowledgement page, I want to do a quick demo and that we are of the software that we have developed that we hope people can use to whether design these experiments or apply to their own data of these lineage barcodes. And because this is still at a relatively early stage, if there is any feedback on what would make sense for the user, that was highly welcome. And you can either give the feedback to me now, or if you can email them to me later, that would be great. So in this app, there are two modes of this little web application that we have developed. So the first mode is what we call the reconstruction, which is to apply the phylotime, which is our phylogeny reconstruction algorithm, and the iSpace, which was discussed today. So the user would start in a reasonable time. I have already run some of these steps. The user would use this upload button to upload their character matrix that carries the mutations in single cells. So if you click this, you can see the format of the input data. You will have single cells labeled by the ID, different cell types, and the different sites that carry mutations. Here, 0 means it's unmutated, and the character string tells the different mutations that are being involved. Right. So after entering some parameters and deciding whether you want to run the algorithm in parallel, you can visualize the reconstruction results. Here, this is showing the results of the character matrix, but it's very hard to see the reconstructed phylogeny here. So instead, we have you can also visualize the phylogenetic tree on its own. Once this was done, you can push the results to the iSpace stage, which along this drop-down box. And here, after inputting some new parameters, you can run our iSpace algorithm to get the fate map topology, and also look at the state assignment along the tree, and plot the distribution of commitment times, as well as population sizes. And at the very end, you can have a summary table that looks like this, that summarizes all the progenitor states that were there, and download them for later analysis. So this application for this type of cases is where you already have the character matrix and then want to apply our methods to infer the dynamics. And on the other hand, if you are planning on doing these experiments and want to design them, we provide an interface to simulate cell phylogenies. To use this, using this framework, you can ask yourself, if I were to run this experiment, what kind of data would I get? And would I have enough sample, for example, in order to infer the particular stage that I'm really interested in? So the input for this step is a table like this, which I probably won't go into detail here, but it defines a number of progenitor states, their parameters, when do they happen, and what is the division rate, at what ratio are the different states committing to the downstream states. But with these parameters input, you can simulate a count graph, and you can make visualizations of how many cell types were there at different stages, and even do a lane plot that visualizes the state transition along the timeline. And given these dynamics, you then can simulate what you would have seen in a lineage tracing. What phylogeny would you have observed in a lineage tracing experiments? given the sampling scheme and sample size that you have. So you would get a phylogeny that looks like this. And then finally, you can simulate along these phylogenies, depending on your specific recording setup, you can simulate what kind of a lineage barcode you would get along these lineages. So for now, we allow only one type of mutagenesis model. We're adding more, including, for example, if you want to read out the somatic mutations during the development, there will be an option for that. So this is not public yet, but if you're interested, I can always send you a version of this to play with. So with that, I will go back to my presentation and show the acknowledgement slide. So we would like to thank people in our lab. So Yi Yang from our lab, student from our lab, is the one who developed the web application, which I was just showing. And of course, our collaborators and funding sources.